Hello and welcome to another episode of Carbohydrate discussion series. Today I will present Nonavalent BODIPY-Mannoside enables lectin recognition. Let's begin the session. Multivalent arrangement of mannosides enables lectins recognition in fluorescence-based assays. So this research paper today I will present, which came into the IISc chemical biology, and they studied the BODIPY with the multivalent arrangement of the α-mannosides. Now, in the abstract, they have studied the three types of proteins, lectins, like langerine, decisine, and Kooyk, in which Kooyk is the plant lectin and others are the human lectins, like langerine and decisine. They have studied little bit, and majorly they have studied the Kooyk. And this type of BODIPY they have used, this type of structure. We can't see here properly, but I will show you in the next slides. So they studied this type of structures, this type of moiety with the BODIPY as a central core, and outside there are the sugars. Majorly they have used mannose. And they studied fluorescence lifetime imaging microscopy, then fluorescence polarization assay, and fluorescence correlation spectroscopies. According to that, they have studied the how it will bind, then what are the other effects, like fluorescence effects they have studied by taking these different types of lectins and this structure. Now, before going further, we just look at it, the structures of tris BODIPY OH, then man9 BODIPY, and man tetraethylene glycol BODIPY. So first, they have already studied the tris BODIPY OH in their previous reports. So further, they moved for the next step, like they conjugated now this 9OH, this tris BODIPY OH having this 9OH free, they glycosylated with the mannose. Now that become the man9 BODIPY. BODIPY is at central and mannose, 9 mannose at the outside core. Now another thing they did, they increased the distance between these BODIPY and the mannose units. For that purpose, they have used the tetraethylene glycol, and they did the same reactions with that, just increasing the distance between the BODIPY and the mannose. So that is called as the man tetraethylene glycol BODIPY. And for that purpose, they have used first α-mannoside 1. And then this α-mannoside 1, further they did the substitution reaction with the BODIPY core. They got the man9 BODIPY. And in second case, this α-mannoside 2 they have used. And the same reaction they did, they got man tetraethylene glycol BODIPY. So these two BODIPYs they have synthesized. Now, before going to further, so what is the objective behind this type of designs like BODIPY sugar conjugate? So first, this tris BODIPY OH, this provides the robust and tunable optical properties because of this BODIPY core. And why the sugar they have chosen? So sugar to improve the water solubility, to increase the biocompatibility, to reduce the cytotoxicity, and molecular recognition for the specific biological targeting. Like for example, Kooyk we are taking the mannose, so specific biological targeting, they have used the particular sugar. Now, let's discuss the synthesis of man9 BODIPY and man tetraethylene glycol 9 BODIPY. So first they started with the tris BODIPY OH, this compound in the presence of pyridine and this acetic anhydride, chloroacetic anhydride they have used in pyridine, DMPA and DCM for 24 hours. They are getting this yield, this compound 6. Further, this compound 6, they did the substitution reaction with this compound 1 in the presence of sodium iodide, triethylamine, and DMF. They got the final compound man9 BODIPY. And in the case of second case, like man tetraethylene glycol BODIPY, they have used this trichloroacetamide, mannose trichloroacetamide donor, and they did the glycosylation reaction with this compound 4, which having this tetraethylene glycol moiety, and this one side there is a thioacetate. They did the first glycosylation reaction. Then after thioglycosylation in the presence of pyridine and acetic anhydride, they got this compound 5. Further, in the case of sodium methoxide and methanol, they deacetylated the compound like all the acetyl groups now become the OH, and this thioacyl group also become the SH now. They are getting compound 2, and these BU3P water isopropanol, they have used room temperature and 12 hour to oxidize the reduce it act as a reducing compound. So in sometimes there are the dimer form like thio compound disulfide bonds has formed between these five. So to break that bond, they have used this second step, and they are getting the compound 2. And finally, the same condition they have used like in previous case, they are getting the compound 9, man tetraethyl glycol 9 body P. Now this was the synthesis. Now first they did the UV absorbance and emission spectra for the same compounds. So spectra A and C, absorbance spectra of compounds in water at different concentration like 0.3 to 10 micromolar concentration they have used, and they studied this spectra, is A and C. And B and D are the emission spectra of the compounds, 1 micromolar of the water and PBS at pH 7.4, and they did the excitation after 520 nanometer. So in both the cases, like man 9 body P and tetraethylene glycol containing body P, they observed the main absorption peaks around 551 nanometer. So here is the main absorption peaks they have observed in both the cases. And upon excitation at 520 nanometer, emission peaks centered around 568 nanometer. In both the cases, you can see 568 here, 569. So this study they have conducted. Now another case, they studied the turbidity analysis. So in the turbidity analysis, turbidity of the solution of both glycobody is increased over time. So in this case, you can see this tris body P OH plus Con they have used. First, this purple line here you can see. Then man 9 body P Con they have used. This red line you can see, and then man tetraethylene glycol body P plus Con they have studied. Here blue line you can see in both the graphs like A and B. What is the difference in A and B? Here B they have used the 50 millimolar of the mannose. In the case of B, mannose means it's not mannose, it's alpha D methyl D mannopyranoside they have used in the second case. Now first case what they have observed, the turbidity of the solutions of both the glycobody is increased over time. According to time, the turbidity of both the solution is increased, but in case of tris body P OH, there is no much change or no increasing the turbidity. And the addition of solution of alpha D methyl mannopyranoside resulted in the fast deagglutination. Now, why this alpha methyl mannopyranoside they have used? It is for the deagglutination process. Means first it undergoes to the turbidity formation. After that, once you are using the mannose, so that undergoes the deagglutination, means that is undergoing to reverse reaction, decreasing the turbidity of the solutions. In case of mannose, so whatever the Con is there, that will again bind to the mannose unit rather than the our glycobody P. And in both the cases, you can observe no change in the absorption was observed in the tris body P OH. Here also it is constant, and here also it is constant in case of tris body P. That means there is no any non-specific interaction between the Con and tris body P OH. That means mannose unit is specific for the Con. That this data confirm the aggregation was consequence of the recognition of mannose residue on the body piece. So according to turbidity analysis, they have mentioned that. Now next we'll move further like fluorescence lifetime imaging microscopy. Before going to the experiment, we'll just discuss little the principle and how it will help in this case. Like fluorescence lifetime imaging microscopy is powerful imaging technique that maps the spatial distribution of how long a fluorophore stays in the excited state after being pulsed with a laser rather than its intensity. Means it will say that first the fluorophore is at a ground state, and once the pulse you have given, how much time it will be stay at the excited state, that will be calculated by the fluorescence lifetime, which is called as a fluorescence lifetime. Now how it will help, like fluorescence lifetime imaging microscopy can identify changes in the molecular environment of the fluorescent dyes. if there will be some change, like we are adding the Corne, so there will be molecular environment change, so that we can confirm. The changes are not evident in other spectral techniques are detected by the measuring the lifetime. So in this case, it is not giving only the intensity, it will give the time at which time the fluorophore at the excited state. This film gives the intermolecular interactions, protein conformation, concentration of the analytes. Then film was used in the work to directly visualize the formation of glycobody Corne complexes. Now going further, I will just discuss little basic of this film. First, what they are doing, this is the instrument where the pulse light source is over here, detector is here, our sample will be here. And by the computer generated data, we can see the data on the monitor. First, a sample is hit with the short pulse of the laser. So ground states of fluorophores will go to the excited state. Fluorophore absorb the energy, jump into the excited state. Another case, then they return back to the ground state and emitting photons over the characteristics time. So whatever the time is, that is a fluorescent lifetime. It will be in the picosecond as nanosecond and nanosecond. So this technique is precisely measure the time delay between the laser pulse and emitted photons. So that time delay basically they are giving as a data. And these decay times are used to build the image where color represents the time. Now in the next slide, I will show you that color map where the different colors are given, and according to that, we can identify how much time our fluorophore will be at the excited state. So here is the data, and here we can see these color lines. So this blue color to the red color. So red color basically there is 8 nanosecond and blue color that is 0 nanosecond. So according to this, we can identify how much lifetime our fluorophore has. It will present the measured lifetime. Now there are two types of experiments. First, Tricomi body pOH they have studied only Tricomi body pOH, only man9 body pOH without Corne they have studied first, then man tetraethylene glycol body P without the Corne. So here you can see in without Corne there is no much difference in the image. Like here you can compare with the lifetime, fluorescence lifetime. Another case, they mixed the Corne with the Tricomi body pOH now. Still there is no much difference in the binding or in the color. Then they have used the Corne plus man9 body P. Now you can see little difference in the color. So it is this green color. We consider, can see here the excited state at this green line. Then Corne plus man tetraethylene glycol 9 body P. There is also color change. So basically, fluorescence lifetime shifts toward the green and yellow colors, indicating longer lifetimes for man9 body P and man tetraethylene glycol 9 body P. What this and there is no unchanged histogram at the Tricomi body pOH. So that confirms that mannosidated body P exhibit global shift to longer lifetimes consistent with the complex formation. So that forms that gives that there will be something complex formation. That's why there is change between initial without Corne and final with Corne difference is there. Now another case they have discussed, like they studied like fluorescent polarization assay. Now what is that fluorescent polarization assay? First we'll discuss the principle. Principle of fluorescence polarization assay relies on the measuring changes in fluorescent molecules rotation speed when it binds to another molecule using plain polarized light to excite it. Now in this diagram, first there will be the polarized light excitation. If there is fluorescent tracer will be in the fast molecular rotation, then in final result there will be the depolarized emission. That means that gives the low fluorescence polarization. But when we are giving the polarized light excitation to protein bound tracer, there will be the slow molecular rotation because there is now protein is available and there is some binding between this tracer and protein. So there will be the slow molecular rotation. So there will be no much change between the polarized emission. So that will give the high fluorescence polarization. Now how it works? First, plain polarized light excites the fluorescent tracer. Second, there is a rapid rotation. If the tracer is free, it tumbles rapidly in the solution and it will give the rapid rotation resulting in the low polarization. Third case, slow rotation. When tracer bind to the large molecule like protein, the entire complex rotates slowly, maintaining its initial orientation. And finally, it will give the high fluorescence polarization. So this is the fluorescent polarization assay. Now going to the result, what they did now. So the three graphs they have given now, polarization on the y-axis and log C, like concentration of Con, they have given at the x-axis in each graph. First graph belongs to the Tris body P OH plus Con. Second graph belongs to the man 9 body P plus Con. Third graph belongs to the man tetraethylene glycol body P plus Con. Okay. So in all the cases they have studied. In first case, so basically in this case, fixed concentration of body P they have incubated with increasing Con concentration in 3 independent experiments. In the first case, there is no binding. So there will be no change in the polarization. So there will be no binding, as we discussed in the previous slides. And in the case of glycobodies, there are bindings. So there is a change in the polarization. It will goes to the high polarization now in both the cases. So glycobodies with Con, that's showing like man 9 body P showing KD value is 300 nanometer. Whether the man tetraethylene glycol 9 body P shows the KD value at 120 nanometer. Now the change in these KD values like man 9 and man tetraethylene glycol, that shows that linker length also have some effect, like not much, but minor and barely significant effect on mannose recognition by the lectin carbohydrate recognition. So once the linker length is increased, so there will be some changes. So KD values has changed. And they found that man 9 body P exhibits the longest fluorescence lifetimes. Here you can see the longest fluorescence lifetimes rather than the man tetraethylene glycol, and longer lived excited state at higher photostability. So now they wanted to focus on man 9 body P instead of the linker considered man 9 tetraethylene glycol body P. So in the next case, they studied the competitive fluorescent polarization assays. Now how they studied? So first, these graphs are belong to the polarization, inhibition, and activity. Now the same case is there. They chosen the man Con A plus man 9 body P, only one molecule. And now sequentially, like titration kind of thing, they are adding the D-mannose, breaking the bonding between Con A plus man 9 body P. And they wanted to see the polarization difference between these. So now what they did, 0.5 micromolar of the man 9 body P plus Con A 2 micromolar and adding increasing the concentration of D-mannose. It's like a titration. So addition of D-mannose result in the reduction of fluorescence polarization. Here you can see in the first graph, there will be the reduction of the polarization while the increasing the D-mannose. That means D-mannose competitively displaced our probe man body P, they displaced, and KD value of D-mannose came around 1.3 millimolar, which is previously reported. Means in case of D-mannose plus Con A, these are the previously reported values. That means it's completely displaced our man body P. Now another case, fluorescent polarization expressed in many millipolarization units, the relation between the inhibition. So other two graphs are belonging to the inhibition and activity of the Con A plus man 9 body P. Now another one more method they have discussed, like fluorescent correlation spectroscopy. Principle is that fluorescent correlation spectroscopy works by analyzing spontaneous fluctuations in fluorescence intensity from tiny observation value caused by fluorescent molecules diffusing in and out or undergoing chemical changes. Now we'll discuss how it works, like first laser excitation, then fluorescence emission, intensity fluctuation, detection, and autocorrelation. Okay, so what they're doing first, a focused laser beam illuminates very small and diffraction limited volume within the solution or living cell. Second, fluorescently labeled molecules passing through the volume absorb the laser light and emit fluorescence photons. Third, there will be the intensity fluctuations. The number of fluorescent molecules inside changes constantly, or random fluctuation. They will measure. Then there will be detection and autocorrelation of the analysis. Now, what is the important? Data extraction is important. Now, what will happen? If there is fast fluctuation, there will be the short lag time. Means small molecules are moving quickly out of the volume. If there is a slow fluctuation, there will be the longer lifetime, longer lag time. Mean larger molecule or slower movement. This we wanted to consider. These two things are important. Now let's go to the result, what they did. So here is the graph between normalized autocorrelation versus lag time in both the cases. In first case, there will be no Kooyk. In second case, there will be a Kooyk. Okay, two cases are there. This this orange line represents man9 BODIPY, and this blue line represent tris BODIPY OH in both the graphs. Okay, so now binding of tris BODIPY OH and man9 BODIPY to Kooyk evaluated via fluorescence autocorrelation curves. So in the absence of Kooyk, man9 BODIPY shows slower diffusion. You can see here. There is the slow lag time is given over here. There will be the slower diffusion in case of first graph. In the presence of Kooyk, man9 BODIPY exhibit strong right shift. In the second graph, you can see here. So in the presence of Kooyk, there will be the right shift. This line you can consider, this blue line. There will be no difference, and other line there will be the difference. So corresponding to the 50-fold reduction in the diffusion rate. So what this data is given? First, there is a monovalent interaction is reported, which can show the four-fold slowdown. And another thing, this data is showing the 50-fold reduction. That means there are multivalent interaction between the Kooyk and our molecule. So that's why there is the 50-fold interaction, 50-fold reduction. That is not the monovalent interaction, which is already reported. And in the case of tris BODIPY OH, there is no diffusion change observed. That means Kooyk is not binding to the tris BODIPY OH. Another thing is that now they have discussed all with the Kooyk. Now two human lectins they have studied. One is called as the langerin, another one is called as the DC sign. So fluorescence polarization assay successfully captured between both the lectins. Here polarization for both the lectins and percent binding they have mentioned. So nanomolar binding affinities they have mentioned for both. KD values for DC sign is 330 and for langerin there is 200 nanometer. Overall presents the like mannose residues on the body core. So there will be the binding between these two proteins and our fluorophore. These are the binding properties they have mentioned for each case, like Kooyk, langerin, and DC sign. These KD values they have mentioned in this. Now concluding the today's talk, BODIPY dyes serve as an effective scaffold for the multivalent display of the sugar rates. Man9 BODIPY exhibits the nanomolar affinity toward multiple lectins due to sugar clustering on the BODIPY core. Probes are valuable tools for comparative fluorescence-based carbohydrate lectin studies, and chloroacetyl thioether ligation enables efficient conjugation of 9 α-mannosides, which we have discussed in the synthesis part, which demonstrating the versatile synthetic strategy. Tris BODIPY OH act as a universal scaffold, allowing attachment of diverse sugar on glycomimetics with tunable linkers. Multivalent glycosylation combined with BODIPY's intrinsic fluorescence enhances lectin affinity and probe performance. And these glycobodies hold promise for lectin trafficking studies, bioimaging, and therapeutic applications, including pathogen-derived biofilms. Thank you.
Good presentation, Mahesh.
Yes.
I have one question. You go in synthesis slide.
Okay. Yeah.
Yeah. After this 3 and 4 glycosylation,
Yes.
why they use pyridine and acetic anhydride?
Okay. So I was also thinking the same. Maybe this thio part has something happened to thio part, I think, because they did glycosylation. Now there is acetyl group also, and there is glycosylated part also, and they will get the compound 5. So actually, I was also thinking the same, that something has happened to thioacetyl or something.
So they not reported anything about
They,
this one.
they have not reported anything that why they have used the pyridine or
And
acetic anhydride.
how they synthesize this BODIPY?
I think in previously report maybe they have given the synthesis of this
No,
body
my
P.
concern they did this na. Which reaction is it? Double one and this.
Which
For
reaction?
the no, for this third one, this the 3 nitrogen is there na. For this they did forgot the name react this one. On double one, this triple one and azide reaction, which is name?
Click reaction, this one.
Click reaction. So when they are they used click reaction on that and then attach after that they attach with body or before the how they gives they did selectively this one.
Okay, so there are two different parts now. So these two parts are similar, right? And
Okay.
this third part is different. Now, I think initially there will be azide at this point,
Okay.
and these 2 points are different now.
Oh, so after azide they did click reaction with this alcohol. Okay, okay, yeah.
Yes.
Now I understood.
Hmm.
Okay, that much I have question. Thank you, Bhaiya.
Okay.
Yeah, nice presentation, Mahesh.
Okay.
Just I wanted to know that how they are synthesizing this 1 and 2 molecule with sulfur at last in.
This one and this two.
With linker length and with the sulfur at in end.
Okay, so like this one they have already mentioned over here, like glycosylation reaction they are doing. There will be always linker, like one side there will be the thiol, another side there will be the OH. So they are doing the selective acetylation of thiol SH unit. SH will be more reactive.
And
So first they
like
did the glycosylation of this.
and sulfur has any role like they are putting sulfur at last in because of some he wanted to so like spin orbit coupling or have some effect on fluorescence. That's why.
So there is no that much effect on that thing. But if there will be the OH, then there will always a problem that OH will not attack on any this acetyl chloride, or it is not much efficient reaction because
Like
there
they
are
can,
other OH also.
amine also they can use nitrogen.
Yes, they could have used amine also. There is no much problem with that. But thiol reaction is always a clean reaction. Like thiol is much more nucleophilic and it will attack easily on any other living groups. It is much more easy reaction than others.
I thought that they like sulfur has role in some fluorescence. That's why they are going with that one. Maybe that one is also a reason.
But still there is no much role because there is only one thiol unit. Like if you are considered okay, 9 thiol units are coming and all, but they have not mentioned any other thing. Okay, because of thiol, some increasing the things or something. Because already there is a body P. It's too much fluorescent molecule.
Okay, thank you. That's it.
Nice presentation, Mahesh.
Yes.
So I don't have any question, just one small query that in one of the slide you are showing right where they have used this linker length when they are attaching some linker, the KD value is reducing.
Yes.
And yes.
Hmm.
So how actually this is happening? Like always this binding and all it will be happening with manoj only, right?
Yes.
So how linker is coming into the picture?
Exactly, I don't know the exact reason. But they still mention like minor or barely significant effect.
Like
Okay,
in this,
whatever you are saying, it's correct. That Connie is directly binding to the Manos at that outer ring, like outer side.
yes.
So whatever the present in the middle. So what is the use of that middle and distance of middle and that Manos?
No, like whatever we are understanding that is correct or not, like you have They have written here, right? The linker length has only minor or barely significant effect on the Manoj
Yes.
recognition.
Yes,
Then
yes,
what is the meaning of this Manoj 9 BODIPY and Manoj TG 9 BODIPY KD? Both are having almost double KD. One is 120, one is 300.
Okay. So maybe this fluorescent polarization assay. So whatever we had discussed earlier, like polarized light they are doing, and there is a rotation of the molecule basically. So much more fast rotation. So there is a depolarization. Maybe molecule size is now big. Something is
Okay.
that big size and small size because it is dependent on the rotation size. This polarization assay, by the way.
So
So
that KD is like it will be average for all the Manoj present on this molecule. Like now lot of Manoj will be there on the periphery, right?
yes, yes, yes.
So in one case they are closer, in another case they might be little far.
Yes.
Like I don't know, like whether whatever I am getting that is the same thing they are trying to tell.
Sometimes it is depend on the molecular weight, molecular size, because it's a kind of biophysical properties. So it is not just because of the binding, but because of size of molecule, how it is big. So if it is big, then where the this molecule is attaching in the cornea, at which site, which interactions are there. So if the size is low, then which interactions are coming together because cornea size is something particular size is there. So there
Oh,
are many things.
yes. One thing we have missed probably maybe the cornea. Cornea is same only.
Yes,
So the moment the molecular size is increasing, probably less number of Manoj might be getting
in
like
contact.
interacted 1:1.
Yes.
This is something to do with multivalency and all, right?
Yes, multivalent effect. Yes, multivalency.
Okay.
I feel like here they did not get the too much credit difference because they are not getting the much they wanted after increasing
Yes.
the linker length. It's just 2 times. In the multivalent system, 2 times is nothing until
This is
unless they are getting some 1,000 times, 100 times. So that's
Mhm.
why they are telling like it's a barely significant effect because 2 times is nothing. That is negligible effect in the multivalent system.
okay. So this change is actually minor in comparison to multivalent effect. That's why they are.
It's nothing actually. So like increasing the linker length, they thought so like okay we'll get some good multivalent effect, but the way is showing with the 9 body pi attached. Similarly, they were getting manos with the tetraethylene glycol attached body pi also, like glycosides.
Mhm.
Yes, correct.
May has come to the experiment where they have showed the turbidity assay, like previous slide, I think.
Yes.
I I feel like everywhere they are getting the similar result. Uh,
Mhm.
So here the blue one went little up. That is also again negligible only. So the crux of the thing, overall the experiment, there is no difference between the linker length attached glycoside and the without linker.
Yes, yes, yes. That's why they are not mentioning it. Only one that experiment they have mentioned. Other experiment they are not comparing both the things because their initial target
Because
was like that only because they synthesized,
uh,
but they wanted to compare.
yes,
Yeah.
always there will be once there is a linker length with this etheric linker, since there is a just linear linkage. So
Yeah.
they thought that will be flexible, that can accommodate more protein, but it's not happening.
Yes.
Okay, miss. As far I have seen, like all the experiments, either like is both are almost comparable.
In both the cases, yes, both the
Yes.
molecules.
Okay, miss. Thank you.
Yeah. If you enjoyed this session, don't forget to like, share, and subscribe. We'll meet you in the next episode, next session. Thanks for watching.